Thanks. All right, so let's move on to some shallow learning now. Today I'm going to present some of our work on how to make sense of chromatin state data. In general, my research interests sort of span this cartoon here from nuclear organization. How is DNA packed in a cell nucleus, and what kind of functional consequences has that to chromatin states, as in how is the DNA more locally compacted and tagged for different functional roles, as well as all the way down to regulatory genomics. If you care about regulatory genomics, please show up at my talk this afternoon at 3 p.m. at the Regsys COSI. There's another completely different subject. All right, so today I'm going to focus on this middle part, chromatin states, and in particular, one central question that everybody seems to be dealing with these days is that we have one genome shared across virtually all of our cells in our body, yet we have many, many different cell types, cellular states, and conditions. And the question is, how can this same set of human DNA encode all these different regulatory programs across all these different cell types? And one way this can be studied, at least starting to understand it to some degree, is by looking at the way different portions of the genome are essentially packaged in the nucleus, or in other words, are tagged by different sort of little chemical tags on these histone tails. Most of you will be quite familiar with this. If you're not familiar with this terminology, just remember colors. The general idea that different regions of the human genome can be tagged by different chemical entities that turn out to be highly predictive of what the functional role of that piece of DNA in that particular cell type is. So for instance, if you have a lot of H3K4 monomethylation, or orange, you are likely to be some sort of an enhancer. If you have a lot of K36 trimethylation, you're likely to be a transcribed region in that particular cell type. So this is offering us an entirely new layer on top of the human genome sequence in terms of what that piece of DNA might actually be doing in that particular cell type. And we are now in the luxury position that these things can all be measured genome-wide fairly easily with techniques such as ChIP-seq or cut and run, cut and tag, etc. The problem is that it results in something like this, something hideous like this, where you have the genome and then all your hundreds or thousands of different cell types, and then you have all these different chromatin or epigenomic marks that you've measured there. So this could be histone tail modification, it could be RNA-seq data, it could be methylation data, it could be whatever. And the problem is that this is kind of hard to interpret, plus it looks ugly. So what we've done previously is to basically summarize these sort of genome-wide tracks of epigenomic or chromatin mark data into a limited number of discrete chromatin states. We basically do this by utilizing a multivariate hidden Markov model that essentially summarizes all these different patterns across the genome into these separate states. And this allows you to, instead of looking at a genome browser with, in this case, 5 tracks or however many data sets you have, you look at a single track with non-overlapping calls or different states. These states, they correspond essentially to different combinations of these observed marks, and they occur in various frequencies across the genome. Before I go on, any questions on this? The fact that you can essentially summarize lots of genome-wide data sets into an individual track per cell type. Okay, good. So this basically allows us now to summarize this dimension, right? So we go from this hideous 3D cube to something that looks a little bit like this. The genome versus our cell types or epigenomes, and basically every color here corresponds to a state here. So here we might see some red, indicating that that might be a transcription start site. And I apologize for this nomenclature here. Here we see some yellow, indicating that that might be some enhancer regions. We see some green, indicating that that region might be transcribed across many or all cell types. What's interesting about this type of data set is that, of course, that it hugely compresses the amount of data you need to deal with, but also it gives you a really good idea of what the dynamics of these chromatin states are between all these different cell types, especially if you start amassing hundreds or thousands of different cell types here. So you see that some regions are fairly constitutively expressed, whereas others are much more cell type specific. Now, there's a couple of problems we have with these things. One is, this is a very small portion of the genome, and the genome is fairly large. This is less than 1% of the genome. But also, we're still generating more and more and more data. So this dimension grows, and this dimension is already pretty huge. So given a matrix like this, for the entire genome across thousands of cell types, where do you start to look? So essentially, we basically need new ways to navigate these type of maps. So taking a little step back here, if you look at a little cutout of this matrix, it's basically an alignment of a bunch of sequences where every element in the sequence is just a color. So here you have a lot of red colors or red states that are aligned, a lot of green stuff. And this kind of looks like what we would look at for an alignment of, in this case, c-MYC binding sites, right? So these are all little nucleotide sequences encoding different variations of c-MYC binding sites. And we never show these things to our family and friends, and we never put these things in papers anymore. Instead, what we like to do is we encode them into these so-called logos, right? So can we now essentially use the same method that we've used for years to take this alignment of sequences and transform it into something like this, to take this and also transform it into something like that, okay? So there's a lot of great theory out there, information theory-based methods to basically do this step, and that's essentially what we did. So I'm going to give you a small example on how that would work. This is, again, the same matrix I showed before, stretched out a little bit. So you have a small portion of the human genome, and you have hundreds of different cell types here. I'm just going to compress it a little bit. So this region is, I think, 800 kb. And if we zoom in to this little region here, which is about 80 kb, we see it kind of looks like this. So we see across all these hundreds of cell types, there's some red stuff, some transcription start site going on here. There's some transcribed regions here. There might be some enhancers here, but overall it still looks a little bit messy. So considering this, this could be just an alignment of different sequences, we can turn this into something like this. And now, on the y-axis, you have a measure of surprisal or saliency or information content that basically tells you how unexpected it is to observe a certain combination of these chromatin states at that particular position. So this is a highly informative region here because it actually lumps together a bunch of these little green states that turn out to be quite rare in the genome in general, okay? So this is a transformation we can, of course, apply across the genome. So we can now take, in our case, 127 different of these chromatin state tracks, compress them into a single Epilogos track, and essentially start scrolling around the genome and immediately being able to hone in on what are the most informative regions of the genome here. This is just a movie, but we've spent a lot of time recently making this actually available in a usable format. Turns out genome browsers are overall, unfortunately, fairly slow, especially for these amounts of data. The real solution came from HiGlass. This is a view of our current prototype on this. You can see the chromatin states here across all the different cell types, and the Epilogos summarization view of this. You can seamlessly pan and zoom around the genome, finding regions of high interest or high information content. You can switch to different genome assemblies, different chromatin state models. There are some readily curated regions that we know have high information content, so are presumably interesting for downstream analyses. We allow you to download, export all the data, either as raw data or as an SVG if you want to use it in a paper or a slide figure. So this is now available at epilogos.net. Like I said, it's powered by HiGlass, which is an amazing browser. It's essentially an entirely new framework for browsing huge amounts of genomic data, initially developed for primarily high-C type of data matrices, and we've essentially repurposed this together with the HiGlass team to work with Epilogos. All right, so just to remind you, we're looking at almost a megabase of data here, which is quite unusual in the genome browser typically. Okay. So this is the general gist of the Epilogos method, right? We go from a huge amount of data where this could span, tens to hundreds of epigenomic marks, hundreds to thousands of different cell types, and by learning chromatin states and then summarizing these into Epilogos, we essentially turn them into these 1D tracks that we think or hope still contain most of the relevant information of our data. So what can we actually do with this, apart from just browsing around the genome and looking at your favorite gene of interest? Well, one thing we can do is we can do systematic comparisons between different cell types of interest. So let's say you're interested in finding regions in the genome that are very different in, let's say, embryonic stem cells versus other cell types, right? In terms of what their epigenomic marks look like. So what you can do is you can simply take these embryonic stem cells, which are these first couple rows in this original chromatin state matrix, and the rest, and you learn Epilogos tracks, or you calculate Epilogos tracks for each of them, and then just do a systematic comparison at every position in the genome. Very simple. One region that jumps out here is this region, which is mostly poised or inactive in ES cells and highly active in other cell types. This is centered around BTG2, which is an anti-proliferation gene, which makes sense in that regard. Now you could say this is fairly obvious because you already see that these regions are very different here between these first couple tracks and the other tracks. But of course you could do this for any kind of arbitrary subset of cell types or epigenomes. So let's say here we're going to compare red versus blue, and we do this across the genome, and we find that this is the readout of where the most differential regions are between the two groups. Does anyone have any idea what kind of cell types or epigenomes or sample properties we are comparing here? So we find that essentially the vast majority of the differences are in the X chromosome. So basically what we've done here is we've taken all the male donors and we compared them to all the female donors, right? So what that would look like is something like this. Here you have hundreds of epigenomes here. Some of these are male donors, some of these are female donors. If we split these up into male and female, we see around the exist locus here, which is the high scoring region there, a very striking difference, as in no activity in male and clear exist activity in female as expected. Again, don't take my word for this. Go to this website, scroll around the exist locus, select the paired option here, which gives you male versus female donors. And here you have a track for male donors, nothing going on there, all quiescent activity. And in the female donors, you see that there's this very clear active transcription start site and transcribed region. You can now zoom out and go, pan and zoom to find other differential regions. And the power of high glass, again, is shown here by this view, which is the entire human genome. And you see all the differential sites here. So this is male donors, female donors, and the difference. And again, you see that all the real large differences are concentrated on the X chromosome. Okay. So these are some of the potential uses for Epilogos. One, the interactive visualization I've just shown you, and I've also shown you some of the comparative epigenomics things you can do with it. Another very interesting application area is that you can now actually learn these little mini epilogos from a single cell type of interest. So let's say you have a single cell type of interest and you have a number of genomic regions that you're interested in. For example, you've identified these regions to be putative enhancer regions, that you can essentially search for these locally recurrent epilogos patterns, just like you would, for example, using meme for sequence motifs. Take that little motif or the little logo and scan a completely unrelated cell type and find concrete examples of these new logos. All right, so, and I'm happy to talk about any of these possible applications with you offline. I think I'm out of time. So for acknowledgments here, a lot of people worked on this over the course of several years in different phases of the project. Alex Reynolds is mostly responsible for the current implementation of this website. He's been putting in a ton of work recently, along with help from the high glass folks, and most notably Peter, Danielle, and Azar. Ting Wang from WashU provided a lot of support earlier on, a couple years ago, when all of this was implemented in the WashU epigenome browser. It turned out, for our purposes, high glass scaled a little bit better. This is work that was initially started in Manolis Kellis' lab at MIT. And yes, that is it. Thank you very much. You could use this microphone if you like.
I'll try to be as loud as I can. Okay, thanks. Thanks very much. This was a wonderful talk. I really loved it. I was wondering, when you are doing the browsing, I mean, this is exploration, right? But, you know, what do you do in such a massive data set? So wouldn't this also be possible, especially if you do the comparison, to maybe even invent a third track that highlights, for example, for some statistical measures, when you compare to Epilogos, say, the sites that have maybe a statistically significant difference, to actually identify then the sites that you wouldn't maybe browse because you know already of the exist locus or whatever, to find something new?
Okay, yes, so there are, so I did not have time to go into this. We have different metrics of information saliency, if you will. Some of them take into account more of the pragmatic expected things, which could be the fact that something is an exist locus, for example. But more importantly, if you go on the website, there is an exemplar region menu that already takes you through the top 100 or 1,000 or something top scoring regions. And of course, we can load that with any kind of list of top scoring regions based on whatever metric. Currently, we have 3 different metrics implemented. So if you want to play around with it, you know, that's something you can do, and you'll see that the list of differentially scored regions are going to be very different depending on the metric. Yeah.
Hey, Wouter.
Hey, Enrico.
Great talk. So as we accumulate more and more data of this kind, do you think we need at some point to revisit the definition that you have of these chromatin states, or at this point you believe these are very robust and don't need to be updated?
Oh no, of course not. No, everything should always be updated. I think so. Another thing I may have mentioned in the split second is that we are actually providing 3 different chromatin state models of different depth, ranging from 15 states to 25 states. The 25 state models are based on imputed data because we simply don't have enough data yet to build these from only observed data. But as we go forward, we will. And I also imagine that we will have, you know, different chromatin marks that turn out to be much more informative than the ones we're currently looking at. I think it's kind of a trade-off, right? I mean, models can definitely get better as you add more and more data, especially if the data are complementary to what's already in there. But it's also really good for people to be able to interpret a model if they see the same model be reused in different papers, right? So you, like right now, this color scheme is probably familiar to quite a few of you because you've seen it in different papers that utilize the data. So I think there's a little bit of a trade-off there, which is why we provide models of different complexity to deal with this. No, there are various theoretically very good ways of doing it, and the typical number of ideal states is either 2 or 8 million. So pragmatically, it boils down to how much of the biological complexity do you want to capture, and how interpretable do you want the model to remain. Yeah. Sure. Yeah. So the question is what distribution and what metric are we using to calculate the saliency score, the information content? There's various ways of doing it. The basic model that I'm showing you here is simply a relative entropy. So it's just simply a Kullback-Leibler divergence, where we take into account the genome-wide distribution of the chromatin states. Some of them are very rare, others are very abundant. But we also have extended metrics, these are also implemented here, that take into account the expected co-occurrence of different states across different cell types. For example, promoters tend to line up at the same position, whereas enhancers tend to be scattered around, as well as similarity between samples. If, let's say, you have 90% is blood and 10% is brain, you don't want to overweigh the blood samples versus the brain samples, and that's something we can take into account as well. The problem is those models get more and more complex, it takes longer to calculate, it gets a little bit more annoying. And for most people, this is a good enough intuitive visualization, but we also provide the others for more deeper understanding. Yeah.
Okay, so
thank you.
Let's thank our first role speaker. We have our first Can I ask each speaker, each of the next speakers to come up during the questions for the previous speaker, just so we can have a quick turnaround.
Okay, hello everyone. My talk is not so related to the visualization. So it's about the data analysis. So just hope you enjoy the 10 minutes. I'm from the Technical University of Denmark, and I'm here to present our package. It's about the gene DMR to calculate the DNA methylations in the gene-based regions. And the epigenetics is a very important field, and it's about the no change in the DNA sequences, but it has the functions changes and also heritable. And the DNA methylations is the major about the epigenetics, and is in association with the health, immune, and growth and reproduction traits. And we can see in the picture, it's adding the CH3 in the cytosines as the epigenetic modification. And if the cytosine followed by the guanine, it's called the CpG site. And there are also some definitions about the CpG site. Is called the CpG island, and also the island shows is regions. Okay. Yeah, okay. It's regions. We can see the. Okay. Okay. We can see the CpG island is a region with the 200 BP, and the GC fraction is bigger than 0.5, and the ratios of CpG is bigger than 0.6, and the shows is just a region with 2000 BP beyond the CpG island. And how to sequence the DNA methylation? There are several methods, and for example, the bisulfite conversion is to dominate the unmethylated cytosine into the uracil. And after the conversion, the unmethylated cytosine will be converted to the uracil. After the PCR amplification, we can see the uracil into the train. So we can know which cytosine is methylated and which cytosine is unmethylated. After this, we can calculate the DNA methylation percentage by the numbers of methylated cytosines divide the total number of read numbers. For example, in the figure, we can calculate the methylation partitions into 16s and also 100. Now there are several our packages can calculate the DNA methylations. They can calculate differential methylated cytosines or differential methylated regions. Some can calculate the DMRs of the gene, also the DMR of the promoter or CpG island. But the package can calculate the DMRs of the interactive regions between the gene bodies with the CpG islands are very rare. So here we provide a new R package, gene DMR, to calculate these interactive regions. This package can input two kind of files. One is the methylation files from the biomarker software or other similar methylation files. And another one is the bed files downloaded from the website. It contains the gene information or CpG island information. then the package can do the quality control, methylation mean calculation in the different regions, and then do the statistical test, and then filter the significance regions, and then visualize the result. And in calculation, the methylation means can in the different regions, for example, from the A to the G, and also the interactive regions, also the whole regions like the window, also the whole genes. But for some regions like the G, there are no CPG sites located in this region, so this region is make no sense. It will be discussed. And then do the logistic regression model to calculate the p-values, and then adjust it to the q-values by the different methods. So we use the real data downloaded from GEO to test this package, and this GEO data is in 2 groups by 5 samples. It's mass samples. And we can see it can output the significant regions, for example, the difference of methylated genes. And it can output 2 different criteria for the filtrations. One is the Q values, another is the methylation difference. And it can also output the hyper or hypermethylated the DM genes. Then it can output the circular graph with the information of chromosomes, the DMGs, and the gene densities, and the CPG island densities, and the show densities, and then with the difference or different methylation levels in different groups. And the most important one, we can enrich the DMGs, the significant genes in different good terms and pathways. And this can visualize by the different categories. If we have one more files from the difference of expressed genes, the DE genes, we can visualize in the 2 categories. For example, the down-regulator or up-regulator in the hypo or hyper-methylated genes, so we can know which genes in the different good terms or pathways. So gene DMR is a user-friendly R package, can calculate the methylations in different regions. Also, there are interactive regions, and it can visualize the DMGs in the different categories for the good terms and pathways. So I need to thank the PhD project of my university and the CSE scholarship from Chinese government, also my co-authors. And also thank you for the attention. And this package is freely in the GitHub. So please enjoy using it. Thank you.
And as we have time for one question, can I also ask the next speaker to stand up? Do we have time for one question?
Hi.
Hi.
Can you hear me?
Yeah.
Did you think of some reasonable way to benchmark your package against the competitors, against some ground truth, whatever?
Sorry, once again. Can you compare your? Oh, okay. Yes, I compare with the MathKit package. Oh. I compare my package with the MathKit package. This package can calculate the difference of methylation situations in size. It's very good in this point. My package is focusing on the interactive regions. It can combine the gene body information associated with the CPG islands. So it's similar, but in different directions.
Thank you for that. Sorry
Yeah,
for
yeah.
cutting off that question.
Yeah, I'm sorry for the microphone.
It was perfect. So thank you again.